All cells generate reactive oxygen and reactive nitrogen species all the time. The electron transport chain is not 100% efficient, so there is some leakage of superoxide anion from the mitochondria as a byproduct of normal metabolism. We have lots of enzymes, oxidases and dehydrogenases, lipoxygenases and prostaglandin synthase, which generate lipid peroxides as well. And oxidative stress is the condition in cells and tissues where the formation of reactive oxygen and nitrogen species outweigh the ability for our cells to protect against the effects. We are out of balance with regard to the production of free radicals and our cells' ability to process them, which results in diseases. Fortunately, we have the capacity to protect ourselves from lipid peroxidation and reactive oxygen species. From the point of view of nature, it doesn't make sense to develop a process that's toxic to ourselves if we don't also have a mechanism for detoxifying and protecting cells. This is an image of the various mechanisms for protecting ourselves from lipid peroxidation, including glutathione peroxidase, superoxide dismutase, antioxidants, including beta-carotene, vitamin E, and C.
The chain reaction of a free radical begins with the formation of a carbon free radical from a fatty acid reacting with oxygen, which here is called a lipid peroxide. It reacts with another fatty acid to form a lipid hydroperoxide, and then the original radical is regenerated with the propagation of an additional radical.
Okay, these are steps of lipid peroxidation. There is some initiating event, UV light or products of metabolism that create these carbon-based radicals, which combine with oxygen to create these other radicals, reactive oxygen species. Those are pretty unstable, and in a best case scenario, they'll react with some antioxidant, which will pick up this extra electron to become less reactive spent antioxidant. So in the case of vitamin C or beta-carotene, it'll pluck off this guy's hydrogen, and oxygen ends up with the negative charge and the extra electron, and oxygen is happy with that extra electron because it's a very electronegative atom. So then we've got this less reactive spent antioxidant that either gets regenerated or gets excreted. And then we end up with this hydroperoxide, which is a little bit more stable than that guy and less likely to do cellular damage. However, if that doesn't happen, then we have propagation of kind of this chain reaction in which then we've got a polyunsaturated fatty acid, like in our cell membrane, and the radical here will react with one of the double bonds here to pull off one of the hydrogens. So this guy, hydrogen, the double bonds go there, and then we've got this ugly, creates another carbon-based radical like we saw up here at the beginning, and the cycle propagates itself. And so we end up with another one of these hydroperoxides, or hydrogen peroxide, which then like to split because the oxygens don't like to be next to each other. It's not very stable, and we end up with two of these reactive oxygen species, which go on to react with other cellular proteins and fatty acids.
Yay!